what we're going to do is I'm going to tell you a little bit about the background about the lab and what we study in the Facette lab. And then after I talk for about 10 minutes, Laura is going to take the reins, so to speak, and tell you about a very specific story in what we learned about a specialized cell type called subsidiary cells and their function. So I suspect that most of the attendees know what stomata are if they're at a webinar for PP systems, but just in case you don't, this is the quick textbook review of what a plant stomata do. And so this is your typical version of a stomatal pore. The stomata is made of two guard cells, which have this characteristic kidney shape. And on the epidermis of leaves, these guard cells allow for gas exchange. So this means carbon dioxide goes in for photosynthesis and water vapor and oxygen comes out. Controlling this flux of gases is really important for plants so they can actually manipulate their stomatal aperture. So when the stomatal pore is open, diffusion happens quite freely. And then when times of stress, perhaps drought stress when you want to conserve water, or perhaps pathogen stress if there's a bacterium that's trying to crawl into this little hole, or perhaps during times of darkness when the plant doesn't necessarily need CO2, the stomatal pore will close. And this is because these guard cells lose their turgor. So it turns out that stomatal architecture has a lot of variation. And so this very common textbook example of two guard cells, which is shown here, that I already showed you, is often seen in textbooks. Sometimes you'll see these other diagrams and sometimes not. But in addition to the two guard cells, which are always present in stomata, there can be some accessory cells, which we call subsidiary cells. So in this plant, in this example, there's actually 4 subsidiary cells, 1, 2, 3, 4, on either side of the two guard cells. And in the case of grasses, they have 2 subsidiary cells here, 1, 2. Grasses have very unique stomata. So in the previous examples that I've shown you, the guard cells are these characteristic kidney shape, but in the grasses, we have what's called a dumbbell-shaped guard cell. And so this means there's bulbous ends, which also can be shown here in a cross-section view, and then a more thinner intersection, or the handles of a dumbbell, shown here. Okay, so there's variation in stomatal morphology, but even though there's this variation, the function of the stomates remain the same. So just like these two kidney-shaped guard cells allow gas exchange, the stomatal pore in grasses is exactly the same. There's a pore that can be open or closed to facilitate gas exchange. Okay, so I want to talk just briefly about these subsidiary cells. What we know is that the way that guard cells open and close is by controlling open and close the stomatal pore, is by controlling their turgor. So when guard cells are turgid, they're full of water, they're full of ions, and the pore is open. This is also true in grasses. So in the bottom row here, we have maize, or another grass, wheat, and you can see that the guard cells here are full of potassium, full of chloride, or full of water here. And you can see the back of the guard cell, I'm tracing here with my pointer, is full of water. What was noticed many, many years ago is that the subsidiary cells seem to exchange ions and water with the guard cells. So in the upper panels, the stomates are closed, and here you can see the subsidiary cells are full of potassium, the subsidiary cells are full of chloride, or here the subsidiary cells are full of water, shown here, whereas the guard cells are now flaccid. Okay, so even though we have observed for many years that these subsidiary cells and guard cells exchange ions and water, the exact mechanism of how this occurs has been a little bit mysterious. So one of the reasons why we study stomatal function in grasses is because many of the things that we eat are grasses. So 3 crops, corn or maize, rice, and wheat, make up about 60 to 70% of the calories that are eaten worldwide. So that doesn't include animals that then eat corn or rice. That's just people directly eating those 3 crops. In addition to those three crops, there's a lot of other grasses which are really important for human health and prominent in agriculture. So this includes things like barley, things like sorghum, also a lot of crops that are in very water limited areas like millet or teff. And so understanding how these specialized stomata work is a high priority if we want to understand how these stomata function in agriculture. Okay, so in my lab, we have two major research objectives. One is, how do the specialized stomata in grasses, how are they made? And the second is, how do they actually function? And so we're not really going to talk very much about how they're made today, but I just want to give you a brief overview of some of the things that we like to do. So if you look at a maize leaf, these are two images from maize leaves, they're shown at scale. If you look very early on in development, all of the cells are sort of these very boring boxes that look the same. And somehow we go from these boring boxes to this beautiful arrangement of cells where you have very specialized cell types, including the stomates. So how does this happen? There's intermediate stages, and what you can see is that there's a very big change from this first stage to this second stage, right? From the third stage to the fourth stage, essentially cells are just getting bigger, but we're very interested in how do certain rows, like this one here, become specified. This row is going to be a stomatal row. How does that happen? And one of the things that occurs at this stage is asymmetric cell division. And so my lab spends a lot of time studying the mechanisms of asymmetric cell division. So the cells that are dividing in this movie are the cells that will eventually go on to become stomates. And so we do a lot of cell biology. We ask a lot of questions about how this process happens, and this means we take a lot of beautiful pictures. So I'm not going to explain all the details here in this seminar, but by all means, you can check out my lab website or my lab papers if you want to know more about this. One of the ways that we study this is we use mutants, and we ask what happens when the stomata are not made correctly. So these are impressions of a wild type or non-mutant plant, and then 5 different mutants. And what you can see in this wild type in yellow are normal subsidiary cells. And I hope that you can notice that in each of these mutants, the subsidiary cells are not made correctly. They're shaped abnormally. And so then we can go on and ask questions about what exactly is broken in these mutants such that you don't get correct stomatal development. And this lets us answer questions about asymmetric division in general. So again, I don't have time to tell you everything we do, but what I can say is that some of these proteins that we know are important for asymmetric cell division have really interesting patterns if you look at them in the cells. So again, we spend a lot of time on the microscope asking questions about what these proteins are actually doing, where they are, and how this leads to different types of cells. Okay, so that's basically one branch of the lab, which is how are these stomata made? And then we have the other branch of the lab, which is really how do these specialized stomata that are distinct in grasses, how do they function? So before I tell you about some of the things we're doing to address this question, I just want to set it up a little bit. And so there's this very nice study from almost 15 years ago now, where they did a meta-analysis and just took measurements from across the literature from about 60 different studies and asked how quickly in different plants do the stomata open or close. And so they measured this time to close, so a small number means quickly, or the time to open, right? And they did this in graminoids, also known as, this is grasses, forbs, those are small leafy plants, and then woody angiosperms or woody gymnosperms. And I hope what jumps out at you is that the numbers here are quite similar, whereas the grasses are very quick. And this is the time it takes for the stomata to close. Reciprocally, again, the time to open, these numbers are relatively high compared to the very quick grass closure. So one of the questions we have is, why are grasses so quick to open and close? They're stomates. And so again, for a long time, people have thought that it has something to do with the unique shape of those guard cells and something to do with the presence of the subsidiary cells. But exactly how this works is still really a little bit fuzzy. Okay, So I'm going to show you an example of some data where we can actually infer stomatal closure. This graph is important for two reasons. The first is that I hope that you can understand why opening and closing quickly might be important. And secondly, La's going to show a lot of these graphs in her presentation, so hopefully you'll understand what we're actually looking at. So on this graph, on the x-axis, we have time in seconds, okay? And then we have two y-axes. One, this GSW, this is how much water vapor is escaping from the stomates. So this is a proxy for whether the stomata are open or closed. And on the other y-axis, we have A, or assimilation. This is how much carbon dioxide the plant is taking up. And the way that we measure this is with a gas exchange meter like the Cirrus 3 or the Cirrus 4 that we actually have the Cirrus 3 from PP Systems. So this is real data that we gathered with one of these gas exchange instruments. So before time zero, when we have our plants in the light, we have some steady state initial rate of both photosynthesis, A, and stomatal conductance, G. If we turn off the lights, this induces stomatal closure. And what you can see is that photosynthesis drops very rapidly. Because of course, plants don't photosynthesize in the dark, right? On the other hand, stomatal conductance, or the amount of water that's leaving through the stomata, this goes down more slowly. And this is because it just takes some time for stomata to close. This is where grasses behave very well. They're quite rapid compared to some other plants. This particular graph is from a maize plant. So what this means is that this time is time where the plant is losing water, but not photosynthesizing. So if the plant is in a condition where it's water stressed, right, which is happening more and more with climate change, this is a potential inefficiency where the plant is losing that precious water that it needs without actually any carbon gain. If we turn the light back on, you can see photosynthesis goes up, as does the stomatal conductance, but the photosynthesis isn't going up as quickly as it dropped. And that's because it's limited by the stomatal aperture. So this delay to maximum photosynthesis is also a potential inefficiency that's limited by the rate of stomatal opening. So the reason why this is important is because for a long time, people have tried to make crops that are more water use efficient. And this turns out to be a little bit of a tricky thing. So there's an idea out there that's been out there for a while and championed by some excellent plant physiologists like Tracy Lawson or Mike Blatt or Graham Farquhar, that basically if you have stomata that are very quick and can respond to the changing environment very rapidly, perhaps you can have a plant that's more water use efficient. So this is an idea that's out there, and there's some data supporting it, but I would say it's still a little bit untested. Okay. So what we wanted to know is what features actually make grass stomata efficient? What correlates with rapid opening and closing, not only within different species, but also within a single species, such as maize. So one of the ways that we're doing this is we're using different maize inbred lines and that have well-defined genetics. So we know what genes are different between them, and also have a lot of variation in phenotype. So these lines have been worked on by the maize community for several years. This is just pictures of ears, which you can see are very different, but there's a lot of differences between these, and we know what their genetic relationships are. So what we're doing with these lines is we're actually looking at the stomates and asking questions about their morphology, their density, how they look different from one another, and also asking questions about how quickly or slowly the stomata can close. And from this, maybe we can get some insight into what makes a stomate close quickly versus open quickly. Okay. Another one of the things that we're trying to do is understand what exactly are the role of these subsidiary cells in promoting stomatal opening and closing. And so we're taking a few different approaches to this, but one of the ways that we've done it is by leveraging these mutants that I showed you earlier that have some abnormal stomates. And so this is the story that Laura is going to tell you today, and I'm going to turn it over to her now.
Okay, thanks for Michelle's nice introduction. And now, as Michelle mentioned, that my lab mates use these mutants with defective subsidiary cell development to study how those subsidiary cell development. And in my research, I use these mutants to study a fundamental question in the field, that what is the relative contribution of subsidiary cell to the stomata function? And if we look at those mutants carefully, we can see that the uh, there are several types of stomata exist. There are some stomata is normally formed in the mutants, and stomata with one abnormal subsidiary cell, and stomata with two abnormal subsidiary cells. So they provide us a genetic tools to study the relative contribution of subsidiary cell to the stomata function. More interestingly, in those mutants, PAN1, PAN2, and PAN1, PAN2 double mutant, the percentage of abnormal stomata complex varies in different genotypes and leaves. So we had a simple hypothesis that leaves with more defective stomata will show less efficient stomata closure. That means that, as you can see here, in leaf 3, leaf 5, and leaf 8, the double mutants always show most severe stomata development defect. So we expect that there will be more severe stomata function defect in the double mutant. And in PAN1 and PAN2 single mutant, they showed in all the leaf numbers, they showed the similar abnormal subsidiary cell percentage. So we expect they show the similar defect of stomata function. So how we measure the stomata function? We have a very good friend, series 3 photosynthesis meter to measure the stomata function. And so in this measurement, it can measure the water vapors from the stomata pores, and then to indicate to us how the stomata open or close. It's also very good to using this instrument because we can control the light and the carbon dioxide concentration exposure to the light. It's also very portable. So you can see here, I can carry it in the field, and I spent a lot of summertime in the field to measure the stomata function in maize. So as Michelle mentioned the stomata conductance figure before, that you can see here, I use this machine to measure the wild type B73 maize stomata conductance, and the x-axis is the time, and the y-axis is the stomata conductance. So at the beginning, I open the light to enable the stomata opening, and at the 0 time, I turn off the light to induce the stomata closure, and you can see clearly in wild type the stomata are perfectly closed. And then I start to measure the double mutants. As expected, that the stomata conductance didn't reduce as B73. And then I applied to the PAN2 and the PAN1 single mutant. As expected, the PAN2 single mutant has lower defect than double mutant. However, the PAN1 shows similar defect, similar stomata conductance dynamics as B73. But we know that the abnormal subsidiary cell in PAN1 do contribution to the stomata function defect, because we see clearly that double mutant has more severe phenotype than PAN2. However, it doesn't make sense to us that, as I mentioned before, that PAN2 and PAN1 has similar abnormal subsidiary cell percentage, but they have the different abnormal stomata function. So as I mentioned before, that we do measure the abnormal subsidiary cells in different leaf numbers. So we want to know if this phenotype is caused by the different leaf numbers. So I use the machine to measure leaf 3 and leaf 5 and leaf 8. We always say that the PAN1, PAN2 double mutants always show most severe phenotype, and PAN2 has more severe phenotype than PAN1. So in these experiments, I use darkness to induce stomata closure. So we want to know that's the factor that lead to the phenotype. So I use another environmental cue, increased carbon dioxide concentration to induce stomata closure. And we saw the similar phenotype as we saw before when we used the darkness to induce stomata closure. And I would like to remind you that when we use the photosynthesis meter to measure the stomata closure, we measure the function of stomata for thousands of stomata. So I want to zoom in to look at how an individual stomata in response to environmental cues. So I use ABA to induce stomata closure, which has been known for a long time can induce grass stomata close. So firstly, I do the test experiments in the light and the light AB condition in wild type, and you can see clearly in the light condition, the stomata are open, and in the light and ABA condition, the stomata are closed. So I quantify this difference of the aperture, which means the distance of a pair of guard cells. And you can see here in wild type, each dot means one individual stomata. And in the light and AB condition, the aperture was significantly reduced comparing to in the light condition. So I applied the same method to the mutants. And also in our mutants, as I mentioned before, that there are three types of stomata with 0, 1, or 2 abnormal subsidiary cell. So firstly, we look at the PAN1 in the stomata with 0 abnormal subsidiary cell. As B73, we can see the ABA can induce stomata closure in PAN1 mutants. And then we look at the stomata with 1 or 2 abnormal subsidiary cell, and you can see that the stomata cannot close even we apply the actual ABA to induce them. And we look at the PAN2 and the PAN1, PAN2 double mutants. Similarly, in the abnormal stomata, the ABA didn't induce the aperture reduce significantly. So that means that abnormal subsidiary cell can cause the defect of a whole stomata structure function. And also we saw a very interesting phenomena in our images. As you can see here in the B73 and double mutant normally formed, you can see the guard cells are very strict. So that means that the mechanical force from two correctly formed subsidiary cell are symmetrically. However, in the stomata with 1 abnormal subsidiary cell, we always saw the curved guard cells, and all the direction is away from the correctly formed subsidiary cell. So it looks like that the correctly formed subsidiary cell can provide a force to the guard cells to enable they close. And when we look at the stomata with 2 abnormal subsidiary cells, the guard cells remain straight. I quantified those phenomena in all four genotypes. We found that around 70% of stomata with 1 abnormally formed subsidiary cell showed curved guard cells, and all of them away from the correct subsidiary cells. So from now that we kind of saw the fundamental question exists in the field that the subsidiary cell is really matter to the stomata function in grass. But we didn't solve the question that why we saw the PAN2 has a more severe phenotype as PAN1. So I look at the normally formed stomata in the mutants, like in the light and light ABA condition. Interestingly, we found that the PAN2 mutants always showed higher aperture than PAN1 in both light and ABA condition in leaf 5. And I repeat these experiments in leaf 8, and we found the similar results. So that's kind of indicate that PAN2 probably has additional function in the normally formed stomata than PAN1. And then I read some literatures to try to understand how does PAN2 work differentially from PAN1. And we found that PAN2 has a one-to-one ortholog GHR1 in model plants, Arabidopsis. And previously, people found it expressed in the mature guard cells, and in the GHR1 mutants in Arabidopsis, it had defect of stomata closure. So we want to know if the PAN2 had a similar expression pattern as GHR1 in maize. So I look at the expression of PAN2 and PAN1 in both development part and the mature part. And as you can see here in the stomata development process, that PAN1 and PAN2 both exist and express very well, which consistent with previous study. And then I look at the mature stomata, and we didn't detect any signal of PAN1 VIP. That means there's no expression of PAN1. But we look at, saw that the PAN2 exists in the mature stomata. So this result consistent with our hypothesis that PAN2 may have additional role in controlling stomata closure, except its development part as PAN1. And we want, so this kind of similar observation in both Arabidopsis and maize. So we want to know if the signaling pathway in Arabidopsis and maize is similar. And previously, study has shown that the calcium can induce stomata closure in both maize and Arabidopsis, and they exist in the very downstream of stomata closure pathway in maize and Arabidopsis. And here is the picture from the Arabidopsis, and they showed that if we provide extra calcium in Arabidopsis, they can rescue the GHR1 stomata closure defect in Arabidopsis. So I want to know if extra calcium can also induce can also rescue the PAN2 mutants phenotype. So I did this experiment, and you can see here in B73, when we applied extra calcium, they can reduce the stomata closure. However, in PAN2 single mutant, we didn't see the calcium can rescue the stomata closure defect. So that means it has the distinct signaling pathway as GHR1 in Arabidopsis. So, as a summary, that our study indicated the necessary function for the subsidiary cell to the stomata function in maize, and we found another protein, PAN2, has an important role in the stomata closure, and we found it's different from the GHR1 in model plants Arabidopsis. So finally, I want to acknowledge my wonderful supervisor, Michelle Facette, who gave me a lot of support from so different ways. And I want to also thanks to the current lab members and the past lab members. Especially, I want to appreciate Dr. Arif Ashraf and the previous undergrad Taylor Morrow, who collaborated with me to complete this project. And I want to also acknowledge all the funding resources who support my research and support my travel to attending the conference to share my scientific research and also communicate with great scientists. Finally, I want to sincerely thanks to the whole PP Systems teams and for Carrie and John for the invitation and very nice organization. And also I want to thank to Bill and all the technical support team. Every time when I had some issue in about the Series 3 machine, they are very responsive and also very approachable and answer my questions as soon as they can. And I really appreciate and help me to do research happily in my graduate life.
Thank you for attending the webinar. Contact us today to learn more about the CYRUS-4 portable photosynthesis system, the 4th generation portable powerhouse elevating the high-level field research experience worldwide.